My friend, yeah, really. Do you have more time than you think?
15 minutes or 15 hours?
Yeah, you have more time. We're early, so.
Excellent.
Excellent.
Well, thank you very much. Today, I'm going to talk about objective one, specifically related to the mother plant. When we initiated this project, we did a back of the envelope calculation and said, oh, how much it's going to cost to produce these plants in a controlled environment? And we can always change those variables to compare in a greenhouse controlled environment or in a fully indoor controlled environment. Right? And then we saw that the limiting factor was actually the amount of space, resources, energy to produce the mother plant. In addition to that, we used some benchmark numbers from the industry then, this is years ago, before we actually wrote the grant, and we said, well, the mother plant is not producing to the level, for it to be moved in a controlled environment where it can be pushed. Right? So then we developed that objective saying, okay, well, let's work on the mother plant physiology and see if we can really push it. And we started a small experiment, and then that gave us another set of numbers. And based on that set of numbers, Mark and I partnered, and this is how he started. Right? So it's been years in the making. So today, instead of my goal is to give you that through PowerPoint slides, right? So I'm kidding. So today, you're not going to see a lot of data. I'm going to let Moin go through the data. But instead, I'm trying to summarize years of work around the mother plant. Right? So you're going to be some plots. You don't want to see standard errors, nothing like that. But hopefully, trying to extract those key points that growers may need. The amount of slides that you're going to have here are coming from the group by NC State, Brian Jackson, and myself, and also from Jennifer. So this image, we actually used it in the grant, and I keep using it because I like it a lot, because I made it, right? So we keep using it to refer back to our objectives, right? So it's the mother plant. Now we're going to talk about two main objectives, objectives 1.1 and 1.2. 1.1 is the environmental optimization, which is the shoot part, right? Everything you can see. And objective 2 is actually the root zone part, everything you cannot see. And then the number one goal of this is to optimize these parameters to get lots of healthy clones and other plants and tips. So the first thing we did, we actually worked on light intensity. And we have 1, 2, 3, 4 different studies on light intensity. We like light in our lab, so we do a lot of light work, right? And the main discovery here was that TPO increases linearly from the levels that we tried. We tried all the way from 10 moles per meter squared per day to 26 moles per meter squared per day, and we have that linear response, right? We didn't want to go any higher than that, especially because then adding more light, it starts becoming cost prohibitive. For two main reasons. Number one, it can be CapEx prohibited if you want to add supplemental lighting, or it's not really just available inside greenhouse conditions. So we stop at 26. But we saw that a response to be linear. So we had 12 treatments, 4 studies, and 3 cultivars that we tested at our light intensity. Right? So let me add a little bit of numbers to that. Right? So all these little dots, it's just a number extracted from all the studies that we did, and try to superimpose it in one graph, right? This will never get published, but it doesn't matter. We're trying to extract the main whole picture information. So here you can see that we were able to increase linearly. And if you increase light between 10 moles per meter squared of day to 26, every mole of light per meter squared per day, it will give you 12 tips per meter squared, right? If you stay within that range. So this will give growers an idea of how much the ability, when they have the ability to measure the light, predict perhaps how many tips they're gonna be able to get. And that may help do planning or do infrastructure investment. Right? So this is live. Please tell me anytime you have any questions, okay? Then we also worked in photoperiod, right? It's important to say it's photoperiod with the same DLI. Before we started this work, we did some reading around flower responses of plants. And we got to learn a lot and how they were classified, and they were mainly classified based on the response in the field, in the geographical location, right? Which there were some limitations for us in control environment that we like to control everything to the T. So we decided to try several photoperiods, And we mainly focus on long day plants, right? And we decided to, based on hypothesis, we decided to say, well, let's work on photoperiod short day, right? Which is a 12 hours standard short day, in plant physiology, and increase our photoperiod. So we found, based on the hypothesis, that increasing the photoperiod decrease the number of tips for that plants. Now we go back to here, same DLI. In order to have the same DLI at different photoperiods, you have to change the amount of instantaneous light intensity. So in order to achieve these 12-hour photoperiods, you need a lot more instantaneous light, that you're gonna have only on for 12 hours. While you're trying to hit these very long photoperiods, then you need less instantaneous light that is present for least number of hours, right? So even though here we have an 18% increase in a shorter photoperiod, it's important for our growers and stakeholders to understand that to achieve the same intensity or achieve that same response, you're going to need a lot more instantaneous light. And that may not be the best economic solution. So we learned that shorter photoperiods, up to 12 hours, give you more tips. So we also decided to test what happens if we go even shorter, right? We wanted to know, can we make this number even higher under shorter photoperiods, right? So that didn't happen, right? So we have that threshold. And again, these are the same DLI. So if you move from 12 to 8.5, that's what we wanted to test. We didn't see that continuing increase, right? The plants start actually reducing the production of tips under the same DLI.
Do you remember which cultivar this was? Was it Albion, right?
This was Albion, this was Monterrey, right? So 5 treatments, 2 studies, 2 cultivars, right? You're gonna continue to see these last names here, where are the students that deserve all the credit for all the work. And at the end, I'll show you a picture of them, okay? So that's photoperiod, right? So these plants, you can actually control the growth of the photoperiod. So how do we take this? How do we take this light intensity and photoperiod into practice, right? So you have your locations of growing, and you have your limitations in photoperiod and your limitations in light. So you can actually play with the numbers and see, where that yield may land based on those limitations. Then CO2 enrichment. We love to work in CO2 enrichment because that's something that we don't really see, we don't smell, we can touch, but it's so important for plant photosynthesis and growth. So some work on CO2 enrichment for the production of strawberry plants. And a quick fact is that actually enriching CO2 in a controlled environment, especially if it's not highly ventilated, is quite affordable. So we decided to look at this because it's a lot cheaper to enrich CO2 than actually provide more photons of light. And so can we use this CO2 to actually increase the yield and the growth of these plants? So we tried different levels of CO2, 500, that's ambient is around 400, but we're working on an inside environment, we couldn't get it below 500, but just imagine this is close to ambient, 500 micromoles per mole, which is the same as ppm. And then we have, 850, and then we have 1,200 micromoles per mole. And we saw that response as well. It's a linear response with the increase of CO2. And we actually attributed that response to a developmental response. I'm showing you here the number of tips for other plants. But what mainly drove this was the number of stolons. Actually, the increase of CO2 increased the number of stolons in the mother plant and then led to higher tip production. We're expecting for more biomass. We're expecting for more tips per stolon, and that didn't happen. We actually have more stolons. But I would not recommend, even though we got this linear equation and we have statistically significant, more CO2, increase of CO2 increases growth. But if you actually look at the numbers closely, the biggest jump is actually from ambient or 500 micromoles to 850, 30% increase on those tips. Now, if you go further than that, even though you keep increasing, we only get around 8.5% increase on those tips, right? So I can even play with the data and analyze it differently and maybe show a plateau after 850. But it doesn't matter. We can do that in the publication. But for the application, we need to focus on if you increase CO2 up to 150, right? And we don't know what happens in the middle, but we know what happens at 150. You will get that big jump in tips and yield with affordable investment. So we have 3 treatments, 1 experiment, and 2 cultivars that we put under this study. Spectrum. There's a lot of work around spectrum, a lot of buzz around spectrum. We do a lot of spectral work in our lab. So we started here in 2020, was published a paper. No, working on that paper was done in 2020, and also more this year. They work on the spectrum. And the main goal was to see what was the impact of the blue in the far right. Right? We know you can use white light, and white light will give you more photons and will give you more growth at a linear response, like I showed you initially. But spectrum, if you want to manipulate the spectrum, these are the main wavelengths that can maybe give you something. So we found actually that if you increase the blue photon flux ratio over 30%, you start decreasing the number of tips. Right? And you really don't see a big impact on plant compactness. We wanted to have the space between the two tips to be very short. So you don't have to have the plant so high up and have those tips closer to each other. We couldn't change that in the plant. That maintained the genetic stability of that was so strong, we couldn't change that like we are able to do in other plants. But we did reduce the yield, right? So in general, we don't want to go higher than 30% blue photon flux. And also, based on Amon's results, we don't see much effect on adding far red to that spectrum, all while maintaining the same intensity of light, right? So that's 6 treatments, 2 studies, and 2 cultivars. Plant density. Here, actually, this is completely Moen's idea, and I'm very glad that he convinced me to go in this direction. So he actually wanted to challenge the amount of plants you can put per meter square in terms of propagation. Right? And he actually pushed the limits going all the way to 18 plants per meter square, right? Typically in the greenhouse propagation, and please correct me if I'm wrong, those of you that are actually growing the plants, but typically around 9 plants per meter square for propagation. Around, Bert, you think, right? So Moen decided to push that based on some early results and see if we can get more tips per area. You can see here in blue number of tips per plant. Obviously, more plants for the same area that are being provided with the same amount of photons are going to produce less tips per plant. That's obvious. This was the conversation I had with Moen. It's going to be lower. And he's like, yeah, but the total amount of tips per meter square is going to be higher. Right? So he was able to show that it was a lot higher. So he actually increased around 64% just by putting the plants together. But the tips didn't look good. Right? The tips, because it was so dense, and we were only working with top lighting, the tips look like crap. I don't know if that's a scientific word. They weren't of high quality. So then he came out with a solution, and I'm gonna let you tell you about it. Right? But plant density was a big breakthrough here on this optimization. So we did 3 treatments, 1 study, and 1 cultivar. Sorry for a typo. So this is an example of the plant densities that Moen put together. You can see good quality plants, and you can see the deterioration of these qualities. And he got a creative on trying to quantify the lower quality that was obvious to the eye, right? So substrate. This work was mainly done by Dr. Brian Jackson and his student Brandon Scher. And the goal of the substrate is first physical properties. This was a test saying, well, there's so many options, so many combinations that you can do with substrate. Which one works best? Right? So it's hard to see, and it's even hard to see for me here. But there is some codes here. You have the industry standard, which is 50% perlite, 25% peat, and 25% core, according to Brian Jackson. Please share if we are wrong on that. And he had 24 treatments in 2 studies, right? And he had mainly 2 main studies where he varied the percent of peat moss versus the percent of core, right? So I have 1 study about the percent of peat moss, and 1 study about the percent of cocoa core, right? So here you can see it's quite busy, right? And industry standard, 100% peat, and then 80% peat, 20% perlite, and then all different irrigations. You're gonna have a copy of this so you can sit down and look at it, and then go back and compare the substrate that you have and see where you land, right? If you're landing over here, right? Maybe there will be a good option to test on site something around here, especially if you're mainly peat moss based. But you can see here, we have products that have wood fiber products included in the mix, right? We have also H pine bark included in the mix. And this is a core-based experiment, and you have a little more success. Don't worry about the absolute numbers, is about the comparison between the treatments, right? Because any greenhouse, the different seasons that make it different. But the comparison between the treatments, you can see here that 80% core versus 20% perlite gave them good results, right? And then you can see how overall the response was quite similar, and they was trying to be a lot better. How about container volume, size, and shape? Right? We know strawberry is a perennial plant. So the hypothesis was, well, these roots behave maybe like a perennial root. Can we give the root more depth for it to explore, or just needs more volume, right? Or it needs a combination of both, right? So Brian and his student actually developed these different treatments. Combination of T is tall, S is short, 3 is 3 liters, 2 is 2 liters. Hope you follow me, because it took me a while to figure this out, okay? So T tall, don't ask me about the 10 and 15, okay? I don't know, but you can see the responses here. The main discovery is that high air space, if you use a substrate that has a lot of perlite, container volume and shape have almost no effect, right? It's a lot more air space there, right? So it doesn't matter if you're long or wide or 2 liters or 3 liters, right? But if you have a substrate that has lower airspace, an example that they made is 20% perlite, then the short container with low volume has lower number of flower plants, right? So this is an example here on when you have high airspace, you really don't see many differences, you know, maybe some separation, but for application, maybe not many differences. And you have low airspace, you can see how actually just the one that is shorter and less volume had a lower performance. Right? So it's just a quick summary, right, of everything that I just told you. I just put it here and made it a little shorter, more concise for you to take home, and we'll continue to work this on the extension publication. We have more coming, and more is going to talk to you about canopy lighting and plant age today, and also a more in-depth summary of what I talked to you about. And this is Sam and Moen. They did the book of the work, and Shan Shu was here before the project doing some of the work. And of course, we have support from the entire lab. So that was mainly focused on the shoot environment. And now we're going to transition to the root environment. And I'm going to have my colleague, Jennifer, to come up here and give us an update.
Ricardo decided that since I was here, I can present the slides that I put together. All right, so my group was focused on nutrient management for mother plant and daughter plant production. And we looked at 3 different factors. We looked at the percentage of total nitrogen that was supplied as either nitrate or as ammonium. We looked at total nutrient solution electrical conductivity, so the strength of that nutrient solution. And then the third part that we looked at was just evaluating nitrogen concentration, with the idea being that we have a recipe that's been optimized for strawberry fruit production in controlled environments, but what modifications do we need to make to that to have a good strawberry recipe for vegetative production? So I'll start with the conclusion, and then I'll show you a couple slides. So what we found is from the strawberry fruit recipe, we need to add a little bit more ammonium to increase daughter plant production. We need to have a little bit more nitrogen because the plants are growing vigorously in a vegetative state rather than a reproductive state. And then also they can tolerate a higher EC concentration. So now I'll show you the data that back that up. So the first thing we looked at was the nitrogen form. And so the standard for fruit production is 90% ammonium, sorry, 90% nitrate, 10% ammonium. And we looked at 3 different cultivars, Albion, Fronteras, and Monterey. And we found that in all 3 of those cultivars, if we provided at least 65% nitrate, which would be 35% ammonium, we had good plant growth. And the different cultivars differed a little bit in terms of where their optimum was. But if we overlaid the response for all three of the cultivars, and we said, if we just look at kind of that, a 95% of maximum yield, where's our range that we can have? And so we can get at least 95% of maximum production if we are providing the plants about 80 to 85% nitrate and about 15% to 20% ammonium. So here's some pictures, and so we can see with Fronteras and Monterey up in the 60, 80, and 100% range, we have good plant growth. The reason why we're not recommending shifting to adding more ammonium is that it becomes then more difficult to manage the pH in our container pots. So if you're willing to either add more lime or be a lot more precise in measuring and potentially adding lime during production, then we can provide the plants more ammonium, but having more around that 80% nitrate makes it much easier for pH management. Then the second part we looked at was nutrient solution, electrical conductivity. For this study, we had two cultivars, Monterey and Albion. And the graphs are set up so that the one here is our control. That's our strawberry fruit production recipe. And so if we increase the EC, we had an increase in daughter plant production. However, as with most crops, once we get too much nutrient solution, then we start to see salt stress in the plants, which you can see on the far right picture. So even though we get optimum or maximum runner production here, we're recommending a little bit lower, just so that we don't have EC stress in the mother plant, as well as in that top layer of daughter plants. So what we find is if we get too high of an EC, we start to get salt stress in these top daughter plants. But the lower daughter plants, the ones that have developed later in the plant, are still great and can root. It's just that our salt stress starts with the mother plant and then starts to progress down the runners. And then lastly, we looked at just fertilizer nitrogen concentration, and the results here were similar to EC, that as we increased nitrogen, we had an increase in daughter plants. But when we were starting to give them high rates of nitrogen, we were seeing a reduction in daughter plant number, because then the total nutrient solution concentration was too high. And then the other thing that we saw that was really interesting, and this is replicated over studies that were done in the winter as well as in the summer across three cultivars, Albion, Monterey, and Fronteras, is that at low nitrogen, we were seeing greater internode elongation on our stolons compared to when we had higher nitrogen concentrations. And then this is also Monterey. And then with that kind of wraps up where I am. So the last study that we have right now is that we're doing an evaluation of the standard fruit recipe evaluated against our best recipes from each of our individual concentration, or best treatments from each of our individual studies, and then combining those all together so that we can get an estimate of an increase in daughter plants when we optimize our nutrient solutions. And so this is the team that has been helping with plant care and management and data collection over the last 3 years. And then top far right is Dr. Erin Yafuso, who was a postdoc in my lab that oversaw most of this research until last fall, when she moved on to a job in industry. With that, I'm done.
Do you guys have any questions?
what hours were you applying CO2 to the plants?
Yeah, we were applying CO2 through the entire photo period, maintaining that high rate. Great question. We know that plants, you know, strawberry plants, they have a very active photosynthesis feedback inhibition. So there may be an opportunity to target that application to be more efficient at the use of CO2. But we haven't done those studies yet. Sherry,
So Ricardo, probably question to Moen, but Flathead was tested but not really effective.
No,
Did you see morphological change?
morphological change, more leaf spread for mother plants, petiole, like longer petioles.
Yeah, but
and
didn't-
more flowers,
Yeah,
which is not.
oh, so more flowers.
More
Oh,
flowers
that's
of
right.
one.
Yeah, that's right, you already got that, yeah, cause we think that Flathead is necessary for food production.
For production.
Yeah, but it makes sense in the running.
In the running.
Thank you, Sherry, for that clarification.
Daniel?
Perhaps this will be kind of tomorrow, but we kind of was saying that you developed some way of measuring plant quality, so like the cracky tips and the good tips.
Yes.
And has that been mapped to field outcomes? I remember one of the students had done some work earlier, showing that the size of the tip didn't really matter that much for field productivity. Sorry if I missed this.
No, perfect.
Well, then I'm fine. But does your measure map to those outcomes?
So we have some quality parameters like morphological parameters, for example. And then like how compact the like unrooted tips, like how productive like unrooted like harvested tips. And like what is the ratio between leaf area there to petiole length, or their greenness, like the span parameter. And we haven't, we tried to root those to see if the change in the quality, if those shade avoidance response affect the rooting of them. And they didn't perform well. They were like so elongated that we couldn't even like stick them in the tray at base scale. But we did not move them to the field.
Daniel, I think it might be looking at two different portions. So I think with Moeen, and I've seen this with another side project that I was working on, is that there might be an impact of the daughter plants that are initially harvested on their actual rootability in propagation. But once you get a plant in propagation, then what's it yield?
I mean, the key question I'm really trying to ask here is, especially Ricardo, you're showing these kind of non-monotonic curves of various outcomes with respect to different environmental variables. And some of those were producing higher quality, lower looking tips, but does that matter? You know, that's just the number of tips coming out. Is that actually marketable tips, or is there another dimension that isn't captured directly that maps from greenhouse
Great question.
quality to actual field?
Great question. Yeah. So the benchmark for us is to produce a good quality tip. That will be market acceptable, right? So we don't, any of the research that increase the amount of tips, if they were low quality, we don't want them, no matter what. Now, we did the size study, which was very, very important for controlled environments, right? In the field, they have a way to pick more narrow sizes differences of these tips to go that eventually will go to the fields. But in the controlled environment, in order to make it affordable, we need almost all of them to be used, right? We need almost every tip to be used. So size-wise, we didn't have time to present it, but we tested the ability to root the entire profile of sizes, and they all root well. And the Mars group took those different sizes, Michael, and put them in the field to see the performance in North Carolina, right? So we solved that, right? But now for crappy trips, no propagator will, by seeing the tips, no propagator will try to make them work even if the numbers are double, because they just, you know, they don't miss that.
I think that's a really good segue, because we can do this, right? Because we're here.
Yeah, thank you, Mark.
So thank you.
Thank you, everybody.
Yeah.